Identification of the hematopoietic stem cell niche and control of the niche size. Hematopoietic stem cells, also known as HSC for short, are located in the bone marrow, which is also known as the blood stem cell. These cells are immature cells that can develop into any or all kinds of blood cells, including but not limited to white blood cells, along with red blood cells, and platelets. Platelets are tiny blood cells that can form blood clots to help stop bleeding. HSC is a cell isolated from the blood or bone marrow that can renew itself, can differentiate to a variety of specialized cells, can mobilize out of the bone marrow into circulating blood. HSCs are the stem cells that give rise to other blood cells. This process is called hematopoiesis. This process also occurs in the red bone marrow in the core of most bones. The hypothesis being tested in this experiment is if there is a role of the basic metabolic panel, also known as BMP, signaling pathway in regulating adult HSC development in vivo. The basic metabolic panel, BMP, is a frequently ordered panel of 8 tests that gives a healthcare practitioner important information about the current status of a person's metabolism, including the health of the kidneys, blood glucose levels, electrolytes, and acid-base balance. This experiment was conducted to figure out what's the role and how significant it is. This was done by testing and analyzing mutant mice with conditional inactivation of BMP receptor type Ia, which is also known as BMPR-IA. Before starting off their experiment, the researchers analyzed the expression patterns of BMPRIA and BMPRIB in bone marrow and surrounding the bone tissue. They found out that BMPRIA was found in the bone marrow, but they were unable to detect the expression of BMPRIB in HCS after their examination. All of the mice were put on different injection schedules. This was to investigate and analyze the HCS population. Although the total bone marrow cell number per femur was reduced in both of the BMPRIA mutant lines owing to reduced cavity room, the number of HCS per femur still increased. The HSC population is a heterogeneous mixture including both of the terms, which is long-term and short-term. They injected the mice with a drug called 5-BRDU to label cycling cells. 3 hours after labeling, they analyzed the HSC population. The percentage of BRDU negative cells in the HSC population increased, and after doing a direct comparison of the entire HSC population, it proved an underestimated but reliable representation of the difference in mutant animals and the control animals. These experiments demonstrate an increase in HSC population, and after 3 months after the injections were done, their blood was examined to see just how much the HSC population was increased, and they found out that it was almost doubled. The HSC population increased significantly after the 3 months. After completing their experiment, the researchers came to a conclusion that there are several mechanisms that can lead to changes in HSC number, one being a natural change in stem cells that either promotes self-renewal or blocks self-renewal. 2, that there's an internal defect that inhibits differentiation, leading to a buildup of stem cells. Or 3, that there's an external influence from the HSC microenvironment. In Figure 1, you can see the expression patterns of BMPRIA and BMPRIB that was found in the blood and the bone marrow. This was done by simply just looking at the blood and the bone marrow of the mice. And Figure 2 is the results of the HSC population increase in the mice. As you can see, the graphs show that. And then in Figure 3, this is just simply the bone structure and HSC population increase, because the researchers found that the HSC population doubled after the injection in the mice. After completing their experiment, the researchers' results were in fact in line with their hypothesis, meaning that their hypothesis was in fact supported, because originally in the beginning they stated that the role of BMP signaling pathway in regulating adult HSC would play a huge role, and that was proven to be correct because the HSC population would increase, meaning the BMP signaling pathway does in fact play a huge role. Also, their predictions were correct too because they stated the exact same thing as their hypothesis, and that they thought that the HSC was in fact going to increase, which it was proven to increase. In conclusion, they simply got mice and injected them with a drug to label their cycling cells, checked them after 3 hours, and the HSC was increased, and checked them again after 3 months, and the HSC was still increasing, meaning that BMP signaling pathway in regulating adult HSC played a huge role.